Hello everybody, welcome to today's COSI Seminar. We have one of these special seminars where COSI students present what they are doing. These are some of the best ones in my opinion. First speaker today, we have Suehyun. She received her bachelor degree in physics from University of Maryland, College Park. She started grad school here at the ECEE department in the fall of 2010. As part of the COSI program, she did a semester rotation with Carol Coxwell, and now she's working with Professor Park on biomedical application on nanorods. So,
thank you. Hi, I'm Suehyun, and thanks for coming to this talk. So just to give you a brief outline of my talk, I'll start with an introduction and go through some background information. And because my research sort of involves biology, chemistry, and optics, I thought it'd be nice to sort of give a short introduction. And then I'll show you some results of our in vitro studies and where we want to go from there. So when an incident light hits a gold nanorod, in our case it's an 800 nanometer or 808 nanometer light, three things happen. One is the excitation plasmon resonance modes lead to highly resonant scattering, and it could be like, in this case, it's the excitation is in vertical direction, but it could also happen this way, and that's why there's another small peak at 500 nanometers. And so there's high scattering due to this. There's also generation of electron-hole pair, which causes nanorods to produce two-photon luminescence. And people have used this to do two-photon imaging. And lastly, there's electron-phonon collisions, and this generates heat. And this one is highlighted in red because this is what we want to use to thermally ablate the cancer cells. And I highlighted this in blue because this is something that we're trying to exploit for simultaneous imaging. And so this is an actual spectrum of absorption spectrum of our gold nanorods. We haven't measured the exact size of the nanorods, but the aspect ratio should be somewhere between 3.5 to 3.7 in order to get 800 nanometer resonance. So the idea behind it is that can we use this heat to thermally ablate cancer cells? And the answer is yes, and it's a great advantage because you can you can locally ablate the cells, and there are two different ways of doing this. One is enhanced permeability and retention effect. I just recently learned about this, and apparently what happens is when you have a tumor, your tumor wants to grow a lot faster than your regular cells, and so it wants to suck up all the resources, and so it has lots of blood vessels around it. And so once you deposit gold nanorods, a lot of it would sort of migrate. If you do like an IV injection of gold nanorods, they would sort of end up in tumors because of this effect without any functionalization. And these guys, von Maltzahn and coworkers, they actually use this to reconstruct a tumor in 3D using X-ray CTs. And this is a thermal image of it. And the second one is targeted treatment, where you functionalize the gold nanorod so that it would only specifically attach to the cancer cells. And this is what we're trying to use. Gold nanorods are great because you can use near IR light, and if you use near IR light, you can penetrate deeper into the tissue compared to other visible wavelengths. Before you just stick gold nanorods into the body, you have to graft a polymer called polyethylene glycol, or PEG. And these PEG grafting has some advantages, and one is that it increases colloidal stability, and this allows your gold nanorods to, it prevents gold nanorods from agglomerating or deteriorating inside the body, and it allows longer circulation time inside the body. And these guys have reported that PEG grafted gold nanoparticles were very stable in blood for in vivo for around 17 hours, whereas before PEG grafting it was like a couple of hours. And then for our case, it prevents non-specific bindings to cells because these guys, so PEG actually goes into, is one of the main ingredients of your soap. And so it's a smooth coating, so it sort of prevents gold nanoparticles from landing in places that you don't want. That's not necessarily cancer cells. And it also prevents body from recognizing gold nanorod as a foreign object. If you just put gold nanorods without PEG grafting, then the body's just going to start attacking gold nanorods, and it's somehow going to be processed out without actually reaching the cancer cells. And so this is an in vivo study done by von Maltzen and coworkers. And as you can see, this is they injected PEG grafted, well, half of the mice with PEG grafted nanorods and half without, and then they irradiated with laser. This black dot is with gold nanorod but without laser. And you see a relative tumor volume decreasing, and by day, I think it was 12, there were no more cancers, and this continued for 2 months. And so this clearly shows how effective it could be in vivo. And these guys used local or enhanced permeability and retention effect, and they didn't even functionalize, and it showed a really positive result. So what we're trying to do is we're trying to functionalize these gold nanorods, which means that we want these gold nanorods to only stick to the cancer cells. And we do this by grafting anti-EGFR. EGFR stands for Epidermal Growth Factor Receptor. And so it's a type of protein found on the surface of all cells. And it promotes cell growth and multiplication when it binds with specific ligands. And just like how there's lots of blood vessels in the tumor, these guys want to multiply a lot faster. And so there's an overexpression of EGFR. And overexpression of EGFR has been associated with 30% of epithelial cancers, including bladder cancer. And so what we do is we attach anti-EGFR, specifically cetuximab, C225 in our case. And once you attach these C225 to the gold nanorod, C225 only specifically attaches to the EGFR. And so for overly expressed if the cell is overly expressing EGFR, then there's more probability that our functionalized gold nanorods would end up there. And so this C225 grafting allows specificity in our thermal ablation treatment. And so this is Huang and co-authors did an in vitro study in 2006. And they wanted to functionalize their gold nanorods, and this is how they did it. They had their gold nanorod, bare gold nanorod, and they put, well, this is a specific PG we used in our lab, but they used MPEG MPG with a thiol group attached to it, and the thiol group sort of directly to the gold. And so they pegylate the gold nanorod, and then they attach the anti-EGFR specifically onto the rod. And they've shown this was a 80 milliwatt exposure, 1 millimeter beam diameter for 4 minutes. And these two are cancer cells, and you see cell deaths right here, whereas this is a positive control and you don't see any cell deaths. And you can tell the cell death by searching for blue cells, and it's a special dye called trypan blue. And if the cell's alive, apparently they spit it back out, but if you're dead, then you can't really spit it back out, and that's why it's blue. We tried using this method to functionalizing gold nanorods, but we've had lots of trouble with it. And one of the biggest problem was that once you centrifuge it, they were like stuck, pelleted, and PG wasn't doing a good job. So in this case, you want enough space so that your antibodies attach to the nanorod as well, and so you don't pegylize the entire nanorod. And that causes some problems, and it has poor colloidal stability. And so we tried 3 different methods. So method A, so all of these methods, A, B, and C, we inserted another step. We added this MPEG-SH, which is, and the thiol group reacts with the nanorod, and so this is just a PG strand. And then this stabilizes nanorod once, and then we add additional bifunctional PG or heterofunctional PGs to create a more thorough pegylation process with the end parts still reactive to C225. And so, sorry, it's probably really hard to see, but this is some of the chemistry that goes on of how the protein attaches to the functionalized or the reactive end of the PEG. And so these are just the, I don't know too much about the chemistry, to be honest. And so one of the question you probably have is why bladder cancer? And I had that question too, like having a bladder cancer in your title slide is not too exciting. But these are some of the reasons. More than 37,500 new cases were diagnosed in US in 2012. But if you look at all these diagnoses, these are 5 different stages. And there's a 5-year relative survival percentage. And most, like 86% of the people who actually get diagnosed are only in stages 1 and 2. And their survival rate is very high. And so if we can treat this if we can treat this cancer, then it's going to, I think it has a great potential of treating people with bladder cancer. And so right now, what they do to, the treatment method that they have right now is called TUR, and I'm not even going to try to pronounce this, but what they do is they basically pour very toxic chemical into your bladder, and they scrape your bladder wall out, hoping that it catches cancer. But then the problem with this is that if you have a recurrence, then there's a really high chance that it's life-threatening. And so if we can catch this, then I think this would be our technology has a great potential. Okay, so this this is a spectrum of our pegylated nanorod, and I just wanted to show slight peak shifts due to polymers attached to the nanorod. And we did a binding assay. In the binding assay, there's a green fluorescence attached to the C225. And so if you show a uniform bright and green fluorescent image, then it shows good binding. This is it's a positive control where there's C225, so it's kind of green all over the place. And this is the conventional method that Huang and others used. This is a new, our new pegylation method, two of our pegylation method. And this PG nitrophenyl carbonate was the best, and the carboxylic group didn't really show much. These are some infrared images. These are bright field images and dark field images. And as you can see, these are nanorod attached, unattached. And there's a bright scattering. And some of the in vitro experiment that we did, this is a gold nanorod treated, and this is positive control. I had some problems processing this data because trypan blue is not definitive. Like live cells there's also false positives as well. And plus this was a black and white image. So I had to actually look through the microscope and check that it's blue before attempting to count all of these to make up power dependence, time dependence plots. I couldn't really, you could definitely see the point where the power gets too much that the regular cell gets irradiated to. But we still need to work on quantification of data. And so what we want to do for future is our collaborators at Angie's campus, they're working on in vivo studies with mice. And we also further want to functionalize it with an upconversion phosphate. It's a sodium ytterbium fluoride, doped with ytterbium, erbium. And this is erbium. You have two ytterbiums here. And what happens is if the ytterbiums accept a 980 nanometer light, they get upconverted to erbium. They get transferred to erbium. And then this rapidly comes down to this state and then emits green light. And so we can change the aspect ratio of the gold nanorod so that it is resonant at 980 nanometers. And then we have been successful with polymerizing UCP, and then now we want to attach that to the gold nanorod so that once you shoot 980 nanometers, you have heat, but you also have green fluorescence so that it could be applied to endoscopes. And that's, I'd like to thank our group, our collaborators in the COSI program, and thank you for listening. That'd be awesome.
You have much better detection right off the transfer.
Okay, because we initially thought about doing flow cytometry, but then our collaborators said that because our exposed area is so small that it'd be hard to tell the difference between unexposed and exposed. But
We have everything.
awesome. Thank you.
I'm curious how deep the light penetrates into, like, a sample. Like, if you're going to do this in a live organism, how deep can the cancer be before you can't do anything about it?
So if I go back to these different stages, if you're in this region, you can pretty much do it with our technique. But because these gold nanorods only attach to the surface of it, you'll have to do it once and then wait a little bit later and then deposit more gold nanorods, and they'll attach the, yeah.
Do you mean you could illuminate from the outside of the body?
Oh, well, that's what you do with a lot of in vivo experiments, but bladder cancer is great because you have commercial endoscopes, and you can just insert it that way. And another thing I forgot to mention here is the advantage of bladder cancer is that you can wash it out. And so once you deposit gold nanorods and they stick to the cancer cells, you can wash out the bladder multiple times so that you don't have ambient killing, unwanted deaths.
Next speaker is Joanna Duncan. She's a fourth-year PhD student working at the Gardner lab in MCDB here in UC Boulder. You will see in her talk that her principal focus in her research is examining novel high-resolution structures on ventral disc Giardia, Lamblia, and its associated proteins. And the primary methods used to complete this project are cryotomography and 3D subvolume averaging.
I was trying to work it out. Oh, can I bring your picture? All right, is this picking me up okay? Can you hear me? No? Yeah, you can hear me? Oh, it's for the video. Oh, okay. So we're going to have another bit of a biology talk today. So I'm going to tell you about one of my projects that I work on. I'm not going to go into the others since this is a 15-minute talk. But I'm going to tell you what we found out about structural variation within the ventral disc of Giardia Lamblia. And first of all, I'm going to give you 3 take-home points. If you don't remember anything else from this talk, try to remember these 3. First one is Giardia Lamblia is a eukaryotic parasite with a large, highly regular microtubule array. And this microtubule array is called the ventral disc, and it's essential for the parasite's life cycle. The structure has 4 regions, which we can assess, we can actually image and access by EM, and they're likely to be functionally distinct. And lastly, these regions differ in their fine structure and their protein composition. So, tell you a little bit more about Giardia. These are what the cells look like. This is a scanning electron microscope image here, and it's just been artificially colored just to highlight where the cells and whatever they're stuck to it. And you can see this is kind of what they're doing inside your intestine. When you get an infection, you'll need to ingest about 10 of these cysts. Now, living in Colorado, you'd probably be warned not to drink the water, especially if beavers are present, because they represent a zoonotic reservoir of these parasites, meaning that the beavers can have them inside them, and they can then infect human hosts. And what they do is they'll basically cover the inside of your intestine and make it really difficult for you to ingest nutrients. So the stage we're looking at here is called the trophozoite. What happens is, remember I said you ingest the cyst by drinking either contaminated water or food. They enter your intestine. Once they pass through the stomach, they undergo a process called excistation. This is where they exit from a thick protein coat that allows them to survive in your stomach acid. They finish a stage of cell division, and then they begin to attach to your intestinal wall. And the way they're doing this attachment is via this organelle called the ventral disc. And I just want to show you this quick video right here. This is a video of one of them swimming and attaching. Now the ventral disc, this is image from the undersurface of the cell, and this thing you can see here, there's some contraction going on as it's attaching to the surface. And these are its flagella that it's using to swim. So let's take a look. These are some images that were made by sectioning through a cell and building up reconstructions by imaging and montaging, and then modeling in the cytoskeleton. So what you're looking at here is the whole cell of the trophozoite form again. The ventral disc here is shown in green, and it's actually composed of microtubules, which are long, thin structures. You've seen these before. Various people have kind of talked about them with fluorescence microscopy. at this seminar, so hopefully you've been attending. But we've just colored it as one solid piece here. And again, those flagella you saw when it was swimming are shown here. It actually has 4 pairs, which are shown in purple, blue, light blue, and this kind of pinky color coming out from underneath. And here's a view from the side. And these brown things here are actually its nuclei. It's a binucleate organism, so it has 2. So what we look at when we image these cytoskeleton, and in particular the ventral disc, are these 4 regions here. We have defined them as 4 functionally distinct regions. So we define the ones 5 or 6 in from the edge as the margin microtubules. We call any ones that are in between there the inner. And then there's this region here where the disc actually spirals back on itself, and we call that the overlap zone. And I'm going to refer to that as the dorsal surface of the overlap zone and the ventral. And that's really what I'm going to focus on today. So let's just take a quicker, little zoomed in view of this overlap zone. So here's the zone again. These are the flagella. One of them actually exits through in between the 2 layers, and these are the microtubules with these unusual microribbon structures. And those are the flagellas you saw going towards the back of the cell this way. Okay. Now, taking a closer look at the ventral disc, I've been talking again about microtubule structures. So there's our model of the ventral disc, our diagram. And if we take this and we flip it 90 degrees and we take a cut-through section, this is actually an average tomographic splice. So this is the kind of data you get from EM by taking a tilt series at plus and minus 60 degrees, so taking many images, using fiducial markers then to do a 3D reconstruction and create a tomogram. And here's the microribbon and microtubule structure. So the microribbon structure extends up to the top of the cell, and this is the microtubule itself. It's quite hard to see when you only got one. This is why we do a process called subvolume averaging. But this is a movie that's just going to take you down through an actual tomogram, showing an example of this data. So what we're doing here is we're moving down through the top surface here. We're just passing through the microribbons of the dorsal surface. You can kind of see there's some connections. Now we enter the microtubule area, and then we pass down into the microribbons of the lower surface, and then through the microtubules. So I'll just let that play one more time. You can kind of get an idea of, this is cryo-EM data. So all these were extracted cytoskeletons that were preserved by plunge freezing in liquid ethane propane mixtures to very rapidly freeze the water or the buffer they're in and immobilize the ice without forming crystals, which is called vitrified ice, and hopefully preserving our samples really well. All right, so this is the kind of information we're getting out of this process. Once we obtain those tomograms I just showed you before on the previous slide, we get to this kind of data. And these are isosurface representations of subvolume average. So what we do is we take our tomogram, like I showed you before, and we'll model along the microtubules. And then at a specified distance, we'll place a point, and we make that the center of a subvolume within our tomogram. And using a program that was developed in our lab called PEAT, we average them together. So then we can, as you saw before, they're quite noisy images. So in cryo-electron microscopy, one of the principal problems is a lack of contrast and poor signal-to-noise ratio. So by averaging many together, we can get representations and find out more about the high-resolution structures. So I'm only going to focus on the overlap zone today because that was the part of the project that I completed. So here's one of the isosurfaces of this is the ventral surface of the overlap zone, and this is the dorsal surface. This one has 7,700 subvolumes, and we estimate to be about a 3.6 nanometer resolution. Same resolution for this, slightly fewer particles. And again, this section is what we call the microribbons, and this is the microtubule. So going in a little bit more detail, again, we can kind of compare the differences in this structure. So when this was traditionally imaged, it was done by 2 different methods, and it was still done by EM, but 2 different methods. One of them was Chem fix, which means putting the cells in a fixative that will make all the proteins condense, and you can see then you get a lot better contrast, but you lose a lot of your high resolution detail. And so nobody knew that this ventral disc wasn't uniform across the whole disc. So now we've shown there's various noble components. We call this guy the side rail, here and here. This is the axis-facing sheet of the microribbon. We have the margin-facing sheet. And this little density is representative of something that you might have caught in the tomogram when I was moving up and down through it, which looks like connections between these microribbons. So because this is an average of many different subvolumes, we think these cross bridges exist, but we're actually, because they're a little bit more flexible, we're actually averaging them out because the position is not uniform in each subvolume. But we're hoping to tackle that later. This is our sidearm. This is where we see one of the most striking differences between this dorsal surface and this ventral surface. And what's so interesting is that we think some of these protein components are the same, but their arrangement is variable along a single microtubule that passes along the spiral of the disc and comes to termination. This structure is called a paddle. This is again where we see a big difference in density here. And we start labeling proteins. GMAP means, G stands for Giardia, and MAP means microtubule-associated protein. So we see several of those, GMAP2, GMAP3, 5, and 7 I've labeled here. And these other ones here, now that you see they're labeled MIP. MIP means microtubule inner protein. These are a relatively newly discovered protein type. So I think it was about 5 years ago they were first identified in Chlamydomonas flagella. And no one's actually ever managed to discover the identity. We can see the density, but we don't know the identity. So Giardia has quite a few of those. So now I'm going to take you through some of what the actual data looks like and show you a comparison between the two surfaces once again. So what we're going to do is just take slices through. So this, you can see this is the kind of data we're obtaining from averages. So we've taken many of those subvolumes. Remember I showed you the single one and it was really blurry and hard to see. So this is why we take an average many together. And I've outlined it to make it more obvious. So then we cut through this section. You can kind of see where we're seeing differences in these inner proteins. So we see this one really strongly represented here. And we think we see more, but in a slightly irregular arrangement on this side. We take a cut through here. I wanted to particularly highlight this because it shows, again, where you see the difference in density on the sidearm here. This has a lot more regularity and a greater number of protein components, we think. Also here, this is MIP5, and this is pretty much my favorite guy on these two structures, because we think we see this in every area of the disc. So we're thinking this protein might be something important, and it might, if we can actually figure it out, we might be the first people to do it, find out the identity of this guy, and maybe it would be a good target for anti-Giardia compounds. Okay, and we'll take another slice through here. Again, you can kind of see the different shapes are appearing. This density is not well represented here at all, but again, here it's much, much larger, and we see this other guy cut through here. All right. So another, if you noticed at the beginning of the talk, I mentioned this work was done in collaboration with Scott Delson's lab in UC Davis. Another reason why we think that these densities are well represented, are representative of different proteins, is they actually see differential protein localization when they tag them with GFP. So you guys have probably seen a lot more GFP than you have EM data, but they're just monochromatic colors, but you can kind of see some localize more like this, and this looks like something that is going to the overlap zone particularly. We see some that localize more uniformly across the disk, and some across the edge. And these are just 3 examples out of about 40 different proteins. So, in conclusion, basically we see fine structures and composition of the ventral disc varies between the 4 different regions. I only told you about 2 today, but there are actually 4. A single microtubule can pass through all 4 regions, and somehow this organism is changing the arrangement of 40 proteins which compose the disc throughout these 4 regions along a single tube. We don't know how, but it's a pretty interesting question that no one knew existed before. Some of these structures appear to be consistent. I mentioned to you MIP5 during this talk, so if that turns out to be a consistent protein across the disc, we might infer that it's important somehow, and then therefore would be a nice target for new drugs that target Giardia specifically. At the moment, the most widely used drug is called metronidazole, and that targets all anaerobes in your body. So as well as targeting Giardia, it's going to target some of your commensal bacteria, which is not the best when you're getting treatment. And lastly, based on the comprehensive GFP localization data, we estimate there are about 40 proteins. And because we know that there's 40 proteins which localize to this disc from our collaborators' work, we're hoping that we can then start tagging some of these with our heavy metal tags and discover their precise location within these structures, which is what we're currently working on. So future directions. Again, I already mentioned the high density clonable tag labels, which we're working with at the moment. And because we know our pool of 40 proteins, it's going to be easier for us to figure out which protein is where in the disc. Hopefully we're going to publish this work shortly, so we just started writing up. And what I really like to do is, using the same data, change the position and the size of my subvolume that I'm picking out within my tomogram to try and home in on those cross bridges and find out their structure. And that's something really novel that no one's ever really been able to image, so that would be great if that works. All right, and I just have my acknowledgements. First of all, I'd like to thank my PI, is Andy Hunger, the whole of the Hunger Lab, so Eileen, Cindy, Mingwa, John, David, Mary, and Sue. And of course, special thanks to Cindy. I didn't show any of her data, but she's the person that imaged the inner part of the microtubules and the margins. So she taught me basically everything about EM, and she's been great. And of course, my funding, which is COSI IGER, and our grant that we have. All right, thank you very much. Any questions? Go ahead. With EM? So at the moment we can't. What you can visualize with EM is protein densities. So when we see a repeating density, we kind of assume that it's the same protein, but we actually don't 100% know if it's something that's, it could be something that's less regular. So if something's irregular, when you average it together, it decreases, it appears to decrease in size. So that's something we'd like to confirm with our methyl tagging, and we're kind of working on that at the moment. We also might try some immunolabeling, which is easier than the methyl tagging because that system requires a lot of optimization. Immunolabeling would give us a much lower resolution information. It would probably tell us if our protein is localizing to maybe the bottom part of our tube down here, at the top, or into our microribbons, because that involves attaching an antibody and then a secondary antibody with a gold particle. And of course, then you're adding about 20 nanometers of distance between where your tag is and where your protein is localized.
Have you thought about looking at it with the scanning microscope to see if you could see some of the structure in the higher resolution?
I haven't tried looking at it with scanning electron microscopy. We have tried imaging a whole cell using FibSem, which unfortunately, by the time it's embedded in the resin and then you cut through with the gallium ion, we're losing some of the resolution. We do see the nice curvature of the disc. And what we're hoping to target with that is actually looking at different attachment stages and seeing how the disc changes in angular orientation of the microtubules to each other as it's in attaching or free swimming. And there's actually 4 stages of change that it undergoes during attachment. And we lose a lot of the high resolution data just from embedding in plastic, unfortunately.
Another thought that may be nonsense is, could you, can you attract are those particles charged, or can you put them in an electric field gradient and attract some of those Giardia?
Can we attract the cells?
Yeah.
Oh, I don't know. I've never tried to do that. The way we culture them in the lab is just, so we culture only the trophozoite form, which is the form that I showed you swimming around. And they just really amazingly like to stick to pretty much any surface we can find. So I've been growing them in various different types of tubes. And as long as you keep their cell media right up to the top and don't let them get exposed to oxygen, they seem to be pretty happy. So.
Well, it would be interesting to see if you could attract them either to an electron with one polarity or the other. With the charged particles, it might be interesting to see if you could systematically collect them.
To see if we can attract the cells?
Either
Or,
plus or minus.
I don't know the response to that.
You can definitely move them around. I don't know if you feel that. There's different things you can do.
Mhm.
Electrophoresis one.
Mhm.
Yeah.
To see what.
That would be another way of looking at it. You've got dipole moments and you get it on and you get induced forces on.
I never thought about that, but yeah, that's interesting. Any other questions? Hopefully that was understandable because I give you like a whistle-stop tour of what's normally a 45-minute presentation. But hopefully you got something out of that. Thanks for listening.
Our last speaker of today is Gardner. He started his higher education at Commonwealth Community College before transferring to Colorado, his department. He received his bachelor degree here in 2010. He stayed at the
Oh, I
department.
took the mic with me. Might work.
He's a recipient of the FTII scholarship, NSA scholarship, a photo minority fellowship, and then a graduate research fellowship. So,
Thanks, Ariel, for the introduction. Like Ariel said, my name is Dennis Gardner. I'm with the Captain Murnane group. And I'm specifically on the imaging team within the group. The group's pretty big and we have different kinds of teams. And I'm going to speak about imaging and the progress we've made towards doing some reflection mode imaging. But before I get into that, I want to just kind of present kind of like a survey of different imaging techniques. This was specifically for biological imaging techniques. But nonetheless, these techniques are used throughout many sciences. For example, one like confocal, your limiting resolution is basically the diffraction limit. And you have, I don't know, say a millisecond timescale resolution for your ordinary kind of standard confocal microscope. Of course, there's super resolution techniques, and I know there's some super resolution experts in this room. That can definitely push down resolution, 10 nanometers or so. And we just heard about electron microscopy, again, pushing down resolution, to a nanometer. Some scopes can go even smaller. But the thing to say with EM microscopy or imaging techniques is that the time resolution is very limited, and same with super resolution. EMs don't really have any kind of time resolution, and super resolution takes seconds. So if we put on here X-ray microscopy, we see that it compares with electron microscopes. They can reach about the same kind of high resolution. That's because the X-rays would have short wavelengths of light. With the short wavelengths of light, we have better resolution. But one advantage of X-rays is it's capable of having femtosecond time resolution. And I'll talk about how specifically how we generate X-rays, we do get this femtosecond temporal resolution. And so with this, we could study some ultrafast dynamics. But one thing that's not mentioned on this survey is elemental specificity. So for example, if we were imaging biological samples, we can say image them in the water window. So this is absorption length on the side. So in the water window where most would want to do some biological imaging where it would be mostly transparent, we could still get elemental contrast due to the makeup of proteins in there. And basically what we're seeing here are the different edges of the inner electrons of different atoms. But this would it's not limited to just biological samples. We could easily be imaging magnetic materials or something like that. And again, we have these different edges of the atoms in which would give us contrast between the different elements. So overall, I mean, the advantages of using X-rays, we could get better image contrast in the water window. We have elemental specificity. We have this nanoscale imaging, ultrafast dynamics. And what I'm going to talk about today is we're working our way to imaging thick, opaque samples. What I mean by this is, say, for example, many companies are producing microchips or something on large silicon wafers. In order to image these, we want to be able to image the surface of the wafer. So we need to work in some sort of reflection geometry because there's no way to image through, an inch-thick piece of silicon. So instead, we need to image the surface, and that's what we're working on. There's many ways to create X-rays. There's two, basically large-scale ways. Synchrotrons. multi-user facilities. You can have a bunch of people around. And there's quite a few synchrotrons, enough that with the proposal so you can get time pretty easily. But then there's also free electron lasers. These are very bright. They can also have femtosecond pulses. But these facilities are big. This is the LCLS. It's 3 kilometers long. Not the whole thing is used, but still very large. Single user facility. There's only one experiment running at the LCLS at one time. There's more smaller, say, tabletop sources, plasma gain. These are good. This is specific technology here is longer pulses than picoseconds. But we do high harmonic generation here. We can get down to attosecond pulses. Desktop size, this is a commercially available high harmonic source. So it fits like this big, fits on a desk. We have high rep rates, we have a coherent source. So I'm going to talk about how we generate our X-rays is with this high harmonic process. We start out with a femtosecond pulse, a high intensity femtosecond pulse that's focused down into a gas. Usually it's a noble gas. And when the laser's in the gas, this kind of highlights, it's like a little highlight of what's going on in the gas, looking at the individual atom. There's a chance that the laser field will ionize an electron. It'll kick off an electron from the atom. The intensity, then the electron's then kicked out, it's ionized. It's going to be accelerated in the electric field of the laser. And then the electric field of the laser is going to reverse, and there's a chance for this electron to then come back and slam back into its parent atom. And when it slams back in, it releases a high energy photon, either an X-ray photon or extreme ultraviolet photon, depending on the gas that we use. However, what we want is this gas. We want a long interaction region right here so that we can get more flux out of the generation. So what we do is we take a waveguide and we fill it with the gas. And by choosing, by a combination of what kind of gas we're using, the kind of waveguide we're using, the intensity of the laser, and the pressure of the gas inside the waveguide, we can balance these different variables in order to get coherent addition of the X-rays. So what we can do is, so as the driving laser is propagating through the waveguide, there's being high harmonic generation throughout this, but since we can match the phase velocity of the driving laser with the phase velocity of the high harmonic beam, and thus we get coherent addition. They're all in phase and they add up together to produce a brighter source. With our sources, then we need to do some imaging. Here's two methods of doing imaging. So some people do diffractive zone plate imaging or reflective mirror imaging. There is no refractive lens. There's not a lens available for extreme ultraviolet or X-ray wavelengths. So instead, but there is a diffractive lens, like a Fresnel lens, which has been used. Reflective mirrors, multi-layer coated mirrors. But both these techniques are limited by the fabrication, so it depends on how well that zone plate is made or how well the mirrors are made. In addition, there's a lot of loss. Putting in the zone plate, for example, there's many undiffracted orders that aren't used in the experiment. So what we do instead is lensless imaging. It's also called coherent diffractive imaging. This is a typical setup of what we have. It starts here. We have the femtosecond driving laser coming into the waveguide. In the waveguide, the high harmonic generation happens. Out of the waveguide, both the driving laser and the high harmonic beam are propagating collinearly with each other. It goes on to a rejector mirror. This rejector mirror rejects the driving laser and reflects the high harmonic beam. Then we go through like a thin piece of metal, 200 nanometers of aluminum or zirconium. In order to absorb any of the remaining visible light, we go through off some multi-layer mirrors, and then we focus it onto the sample. And this is the lenses part. Between the sample and the detector, there is no lenses. There is no mirrors. Basically, the detector just records a scatter pattern from our object. We have this scatter pattern, and with the phase retrieval algorithm, we retrieve the phase of the scatter pattern, and we're able to reconstruct the object. This is just a little diagram of how our phase retrieval algorithm works. We start right here. We start with the scatter pattern on the detector. And we don't know the phase, but we take a random guess at the very beginning. We apply a random guess, and then we Fourier transform into object space. In object space, the first time we go around, it's not going to look right at all. It's going to think there's object everywhere. We supply a support constraint. A support constraint, in this case, being the object isn't infinitely big. It's in some area. So we apply a support constraint, and that gives us a new guess of the object, and we Fourier transform back into frequency space. And now there's a little bit better phase guess here, but the amplitude is going to be not correct. So we keep the phase here, we replace with the measured amplitude, and we iterate again, applying the support constraint again, and we iterate through this. And with this, we have demonstrated, for example, 22 nanometer resolution with 13 nanometer light. So we're not at the diffraction limit, but still we're getting high resolution. This is on a tabletop setup. And what we imaged was a test sample, say. This sample here, it's all opaque, and etched into it is this figure is etched into it. And so it's basically like a little pinhole. It's definitely isolated. It's a nice sample. Even if the beam doesn't look so great, it's going to get basically just filtered by this little pinhole / object that we're imaging. So what we want to do is take a step forward from this, and instead of imaging something that's all opaque with a little pinhole, imaging something maybe that's all transparent and an extended object in a reflection geometry. And one step to get there, or how we started to get there, was using visible light first. So this is done with a Heaney laser, visible light. And the reason we started here was just to quickly develop a prototype, quickly go through the algorithms, develop our techniques, and at the end I'll show how we're transferring what we've learned here into the extreme ultraviolet. So basically, if we have a sample and our sample is much bigger, it's not isolated. Ideally we'll image something on a big wafer, and here we just started with this like a big Air Force test pattern. And so we need instead of having an isolated object, we can have an extended object and then instead isolate it with illumination. So what I mean is we have the laser coming in, it goes into an aperture. There's an imaging system which images this aperture onto the sample so that only a small area of the sample is illuminated. And then it's allowed to diffract onto the CCD. And in this case, in the visible, we had a lens here between the sample and the CCD in order to perform the Fourier transform. So we notice some things here in this reflection geometry. One, which happens anytime you're doing high NA imaging of this sort. And basically, what happens is, the scatter pattern from the sample it's on a sphere, and that is getting recorded by a flat detector. It's the same kind of problem that happens, say, like on a map of the world or something, when you're projecting a globe onto a flat piece of paper. There's distortions at the edges. And so that happens in our scatter patterns. And another thing is, because the sample is tilted with respect to the incoming beam, the incident beam is not normal to the normal vector of the sample. And so this causes a tilt of the diffraction data. And so what happens, the scatter pattern that we're collecting is not linear in frequency space. And so what we do is we correct for this. We remap it onto a linear frequency space in order that we can run our reconstruction algorithms on the scatter pattern. And we know how to correct for this, knowing what wavelength of light we are, how far away the detector, things like that, and the angle at the sample that was. So what we do, we get our scatter pattern, we map it on the linear frequency space, we pop this scatter pattern into our reconstruction algorithm, and out comes a reconstruction of the sample. And so this is a traditional microscope image. Again, this is in the visible, so we can easily use a regular microscope to image the sample. And we took different scan positions of the sample, stitched them all together, and this is the reconstruction with our lensless technique. And so the sample is all transparent. This is a nice piece of glass, a little chrome deposited on it. So what we want to do is we're starting to transfer this, what we developed in the visible, into the extreme ultraviolet regime. So this is just recent data, and this is what we're currently working on now. We have our high harmonic beam. It comes through some multi-layer mirrors to pick out a single harmonic. And we need to in this case, we're imaging an extended sample that's not isolated. So we need a way to isolate it. And again, we're going to isolate it with illumination. And so we pass the beam through a pinhole in order to give it that hard edge. It goes through the pinhole, and then it goes on the sample. And between the sample and the detector, again, there's no optics, there's nothing. So it's just lensless imaging again, and we record the scatter pattern on a detector from our sample. So this is what we get. So without the sample, since it's mostly without the sample we just get the beam. With the sample we just get a little bit of diffraction because it's almost an entirely transparent sample. This is much different than the traditional samples being imaged with lensless imaging. So it's mostly transparent. Here we plot the intensity to the 1/4 power just to see more of the detail in the diffraction pattern. And then we can plop this into our reconstruction algorithm and reconstruct the object here. And this is an SEM of the object we were imaging. And it's all transparent. So this gray stuff, it's all 20 nanometers of chrome deposited on silicon nitride, which is transparent. And then this part, what looks black, is actually even more transparent. It's where the chrome was etched away. And so it's very transparent here and then completely transparent over here. And how this was imaged, it looks a little funny because the SEM also had a focused ion beam on it, and so the actual image from the SEM is at 52 degrees. But the reconstruction agrees well. Okay. So that was one step. So one step is being able to image an extended object. And another step now is we have started collecting data in a reflection geometry. And so we have the waveguide, we get our high harmonic beam, we're reflecting off the sample, and clearly what we're imaging here is a bunch of little nanopillars, like 20 nanometer nanopillars. And there's clearly this non-linear frequency scatter pattern, but we can remap that. And what we're currently doing is we're modifying this setup here, and we're putting in the pinholes needed in order to isolate only a certain section of the sample in order to reconstruct it. But we can get, or what this shows is that we can get enough scatter off a sample in order to collect a scatter pattern in order to reconstruct it. So with our beams, we have high resolution, we have high spatial resolution, high temporal resolution, we have chemical specificity, and we're right now very close to imaging very thick samples. And this is of interest to many companies that pattern things on big wafers. And it's also in general just for science, for example, studying ultrafast demagnetization, acoustics or energy transport. This energy transport and how heat is flowing in tiny structures is going to be very important for the next generation of computers or data storage and things like that. So that's all I have. Okay.
You mentioned femtosecond resolution. Is that potential or real? Because your flux is really low, so you need to integrate over a long period of time. So how would you go femtosecond?
Well, yeah. So typically when we do imaging, we're integrating for half an hour or so. The femtosecond is more for right now at least, say in some of the studies of magnetism, where we have pump probe experiments going on, and we're pumping with the red and then imaging and then getting the scatter pattern, and that we can say have a certain delay and integrate there, and then move the delay and integrate again, and so we're able to get that time scale resolution in I think in reference between the pump and then the probe, and then we integrate it at different intervals there. For imaging, we're not there yet. We don't have enough flux to say do one shot single imaging yet.
Any other question? Okay, so let's finish another